Hi everyone and welcome back to my channel. So today we'll be discussing about the T lymphocytes acting in the acquired immunity. So okay, as you know, in lymphocytes there are three types, right? One is the B cells, another one is the T cells, another one is the NK cells. This NK cells acts in the innate immunity, and BT cells act in the uh, acquired immunity, right? So today, so in T lymphocytes there are certain types, right? In each cell there are certain types. So in T lymphocytes there are the CD4 T cells, and then the CD8 T cells, and the suppressor T cells, and the gamma delta T cells. Okay, so each of these cells performs a particular function. Okay, like the CD4 T cells act as the helper T cells, and the CD8 cells directly go and attack, and thereby it is called as the cytotoxic T cell, which yeah, and then the suppressor T cells, which suppress autoreactive T lymphocytes. These uh, T cells which go rogue and go and attack the host tissue itself. Those are supposed to be suppressed, and that suppression is done by the T cells, these suppressor T cells. And this gamma delta T cell is actually present in the mucosa of the GIT, and it prevents the antigen entry. It somewhat works like the innate immune response. Okay. So as you know, a cell has a type of receptor, right? Just like that, the T cells also have a receptor, and that receptor is known as the T cell receptor. Okay, this T cell receptor has certain constituents, and each constituent has its own particular function. Okay, so first of all, there are these receptors are actually broken down into subunits. The first subunit we'll be discussing about is the alpha beta subunit. You see this alpha and beta subunit. These are actually together bound by the disulfide bond, and over this location only this antigen comes and sits, or comes and binds with. So this is the antigen antibody antigen receptor reacting place. So in favor, or to help this out, there are certain other subunits such as the epsilon subunit, the gamma subunit, the zeta subunits, and all are present side by side. Okay, so as you know, these three, the gamma, the delta, and the epsilon subunits together form the CD3 proteins. Okay, what is CD? First of all, CD is cellular differentiation. Okay, so yeah, and then apart from these subunits, there are others, such as the CD28, which acts as a co-stimulatory molecule. And then the most characteristic feature is the CD4 or the CD8 receptor present over here. See, in a T cell, it can either have a CD4 or a CD8 receptor. It can't have both. So yeah, what is the function of a T cell actually? The T cell mainly mediates, it mediates cell mediated immunity. Okay, and the CD4 positive T cells help the B cells. Therefore, they are called as the helper T cells, to produce these cytokines. Cytokines are these enzymes which goes and attacks these bad antigens. And the CD4 T cells directly go and kill the infected or tumor cells. These CD4 T cells are there, right? They go and act upon abnormal cells. Okay, so what are abnormal cells? Such as the virus infected cells and the tumor cells, which are abnormal. And how does it actually go and attack these antigens or these cells? That is via the perforins and the granzymes. These perforins actually make holes in the cell. If there's a cell, it makes holes in between, and through that the granzymes enter into and destroy the bad cell. And these T lymphocytes actually cause delayed cell mediated hypersensitivity. Cell mediated itself denotes that it is a type 4 hypersensitivity reaction. And this helps in the acute and chronic cellular organ rejection. Not really a help actually. So, as you know, these T cell receptors, these CD4 and CD8, are supposed to bind to the B cells, right? The B cells MHC receptor. These actually, what happens is that this CD4 or 8 should be going and binding with the MHC class receptors. Okay, the CD receptors are from the T cells and MHC is from the B cells. Okay, so in MHC also there is MHC class 1 and MHC class 2. Okay, so what happens is that it is known as the rule of 8. So what happens if there is a CD4 cell, it has to multiply with a number that gives out the number 8, and that will be the number of the MHC class of the B cell. So it should be 2. That means a CD4 positive T cell will combine only with a MHC class 2 receptor B cell, and CD8 positive T cell will only combine with a MHC class 1 B cell. Okay, so yeah, that is it. And then you see all these cells actually are produced and actually matured, right? As you know, the B cells actually develop and mature in the bone marrow itself, but the T lymphocytes aren't the same case. These T cells actually are produced in the bone marrow. These are produced in the bone marrow and matured in the thymus. Okay, so these T cell precursors are there, right? These are actually given or are sent to the thymus, and it is sent in the form of CD4 CD8 T cell. This is actually an immature form, and these immature forms are present in the cortex. As you can see, this is the cortex. This is the cortex region. Okay, and over here is the what is present is the CD4 positive CD8 positive T cells are actually present over here. And then these maturation occurs, that occurs in the medulla. So in the medulla is where the CD8 positive or the CD4 positive cells are actually matured up over here. Okay, and these as you know, the CD8 positive can directly go and attack the antigen, so therefore it forms the cytotoxic T cell, whereas the CD4 positive needs, it can't work on its own, but it can definitely lend a helping hand. So it forms the helper T cell, which helps the B cells to conduct immune responses. And these helper T cells are of various types. There's the T helper cell type 1, T helper cell type 2, T helper cell type 17, and the regulator T cells. And each of these produce certain enzymes, and are promoted by certain enzymes and inhibited by certain enzymes. And these secrete certain enzymes, and they cause some functions, such as the Th1 cell. Its function is to activate the macrophages and cytotoxic T cells, and the Thc2 cells activate eosinophils, thereby increasing and it increases immunoglobulin E. And Th17 induces neutrophilic inflammation, otherwise known as acute inflammations. And regulator T cells prevent autoimmunity. That is, it maintains the tolerance level. Okay. So yeah, as you know, the cytotoxic T cell or the CD8 positive T cell, it kills virus infected or neoplastic cells or else tumor cells, right? Tumor cells and donor graft cells by inducing apoptosis. It induces apoptosis. It releases these enzymes known as, what did I say? Perforins and the granzymes. Perforins add a hole to the cell, and granzyme enters via it and destroys the cell. As you know, by the rule of 8, the CD8 binds with the MHC class 1. And then the regulator T cells. These help maintain specific immune tolerance. It helps in maintenance of the immune tolerance by suppressing the rogue T cells. It identifies the expression of certain CD receptors and regulates it. It may upregulate or deregulate it. So yeah, and now the most important is, how does a T cell actually be activated? For that, there are specialized antigen presenting cells. One of the most common example is the dendritic cell. So where are these dendritic cells actually seen? Dendritic cells are actually seen in locations where our body is in contact with the environment. Okay, with the environment, such as our skin. Our skin is actually in contact with the environment. Our GIT, because it's actually a hole. The GIT, I would say, is actually a hollow space, right, through which food enters. So by that way, actually antigen can also enter into it. So yeah, over these places and all, the dendritic cells are present, which actually ingests and processes the antigen, then migrates to the draining lymph nodes. Because as you know, in the lymph nodes only these mature cells are present. These mature cells are present and are quite important. Yeah. So yeah, and then what happens? The T cell activation, signal 1. What happens is that these exogenous antigen is presented to the MHC class, such as for example, if there is an MHC2, it is recognized by the T cell receptor of the CD4 T cell. Or if it's an MHC class 1, then it is recognized by a CD8 positive T cell. And moreover, our T lymphocytes are actually very shrewd. They need a secondary confirmation. So a second signal, where is it? A second signal is sent. That is the co-stimulatory signal sent via the CD28 and the CD80 or CD60. That is why, as you know, see this over here. This goes and binds to the other molecule present outside. That might either be a CD80 or CD86 molecule, right? Yeah. So this gives a confirmation. Okay, now you can go and attack and go and destroy this pathogenic cell. Then what happens? This activated TH cell produces cytokines. So yeah, and then this enables the T cell receptors and are able to actually recognize and kill virus infected cells or neoplastic cells. So yeah, this is T lymphocytes and how they act upon in the adaptive immunity. So I hope this video was really useful for you, and thanks for taking your time, and I hope it was worth it. So yeah, thanks for watching, and make sure to click the like button and click the subscribe button and click the bell button, and as always, share it with your friends. Let them know and share this information, and I hope you like this. So yeah, thanks for watching, and as always,